Gene expression is fundamental to all life on Earth. It is the process through which information from DNA is turned into proteins and non-coding RNAs that provide much of the structure, function, and regulatory processes in living organisms. In the context of conservation, knowledge of gene expression is central for assessing species health, monitoring populations, aiding reproduction efforts, and making informed decisions to safeguard biodiversity in a changing world. In conservation biology, gene expression studies provide important insights into an organism's basic functions and environmental interactions. RNA sequencing, or RNA-Seq, is one of the most popular tools for understanding gene expression. However, the accuracy of RNA-Seq can be affected by multiple variables, potentially yielding misleading results. To understand this further, a group of researchers from George Mason University, the University of Montana, and the state of Montana conducted RNA-Seq using different types of tissue samples from a non-model fish species, the threatened West Slope cutthroat trout. Specifically, they evaluated the influence of sample processing time, if sampled immediately versus 5 minutes after euthanasia, sampling method, electrofishing versus dip netting, and RNA-Seq library type, whole mRNA-Seq versus 3' RNA-Seq on gene expression estimates. They found that all studied tissue types produced good quality RNA if sampled within 5 minutes after euthanasia. The sampling techniques and tissue processing time did not influence variation in gene expression. This is vital information for researchers working outside of the lab when samples cannot be processed immediately or when flexibility is needed for collecting study organisms. However, when comparing the RNA-Seq library types, whole mRNA-Seq was found to detect 31% more genes than 3' RNA-Seq. This implies that whole mRNA-Seq is a more potent option when detecting a higher number of expressed genes is vital, especially low-expressed genes when alternative splicing is of interest or when working with an organism lacking adequate genomic resources. However, 3' RNA-Seq is less expensive than whole mRNA-Seq, making it much more accessible. For studies where detecting alternative splicing is not of interest or when working with a model organism with plenty of genomic resources, 3' RNA-Seq is a suitable and cost-effective option. Overall, the results indicate that while researchers can use either fish sampling method in the field, the choice of RNA-Seq library type should be determined based on their specific research needs.